Good morning, my name's James Stanger. This is another video in my euploid embryo series, this one looking at sperm DNA fragmentation and euploid embryos. So, by way of introduction, sperm DNA damage or sperm DNA can be damaged during spermatogenesis, during oxidative or environmental stresses, resulting in a variety of damage that varies between individual spermatozoa. If excessive, then sperm selected for IVF or ICSI may contain some degree of damage that the zygote may or may not be able to repair. Identifying if a sample is at risk is difficult just from a semen analysis. It's a big ask. However, it's important to point out that euploid embryos are the current preferred status for transfer. Most aneuploid embryos are due to oocyte meiotic errors. So most of the aneuploidy comes from the oocyte. Therefore, why should sperm DNA impact on the euploidy rate? The zygote has some capacity to repair both sperm and oocyte DNA, but there are time and structural limits to complete the repairs. Viable euploid embryos need to meet very strict development criteria. The sperm DNA may hinder. So the question is, can a level of sperm DNA fragmentation be detected in a semen sample that could relate to or impact on euploidy rates? If so, there may be a better sperm selection technique, such as the new microfluidic devices, be used to increase the probability that the sperm selected for microinjection has little or no DNA damage. After all, one only needs a few sperm from 100 million or so for insemination. How hard can it be? So the two key point summaries on euploid embryos. If you're an IVF staff member, you all agree that euploid embryos are determined after PGTA, post-genetic transfer, or PGTA. They represent viable embryos for transfer and storage. They may either be euploid or minimally mosaic embryos. They're identified by the biopsy of trophectoderm and not the inner cell mass. They may not truly represent the future health of the baby. They do have, though, a fairly well documented 50% lower incidence of miscarriage. And since after transfer, they are most likely to result in a live birth, many studies are now concentrating only on euploid embryos. What can they tell us about implantation in IVF? Is it a new standard? If, however, you're a client of IVF, someone seeking IVF services, then the important information you need to know about euploidy is that about 40% of all embryos that have the correct number of chromosomes, and they're called euploid. And that can only be determined by PGTA, pre-implantation genetic testing for aneuploidy. Euploid embryos have a lower chance of miscarriage, so more likely to go to full term, but some embryos are labelled as not euploid, that is, they're called aneuploidy. They may overestimate the likelihood of the inner cell mass and the baby being abnormal. Some embryos are a mixture of euploid and aneuploid cells. These are called mosaic, and the degree of mixture can reflect the health of the future child and the likelihood of pregnancy. So the purpose of this talk is based on a paper which was published recently, 18th of November, by a Chinese group doing a systematic review and meta-analysis for sperm DNA fragmentation on embryo euploidy rates in IVF. And just by way of background, how the sperm DNA is packaged is important in the testing. And there's quite a good paper here from a 2024 European Journal of Cell Biology looking at sperm chromatin and basically talking about how sperm chromatin is packaged both in terms of epigenetic markers and in the relationship between histones and protamines, and that the package in the DNA is actually really critical, and how the DNA is packaged can make it susceptible to damage. Basically, the highlights here are the sperm chromatin is extensively remodeled during spermatogenesis. Crosstalk between epigenetic signatures, DNA, histones, protamines, and RNA shapes the sperm epigenome. This is a little bit off track. The relationship between sperm chromatin integrity and embryonic development requires further investigation, and that is what this talk is about. Sperm chromatin protamation, condensation, integrity are crucial for reproductive success, and this is also what the talk about is. And the other thing I think is that there are different tests for sperm DNA fragmentation, and basically they look at different things. This is an article published in 2013, almost 12 years ago. It's quite a good one, actually, looking at the different tests and how they relate to fertile and infertile men. There's a COMET test, which is basically both neutral and alkaline, that tests for single and double-stranded DNA breaks. There's a tunnel test to look for nicks in sperm DNA, and the sperm chromatin and SCSA test are basically looking about how well the chromatin's been replaced with protamine and how well that sperm packaging has been put together. And I guess the interesting here, the tables here, is that if you look at the DNA fragmentation range between fertile donors and infertile patients, even within the fertile population, there is about 1 in 6 sperm using the tunnel or SCSA or SCD tests, about 1 in 6 of sperm still have damaged sperm. But this goes to about 1 in 3 when in infertile patients. So basically, I think so when you look at these studies, you need to look at tests being made because they're looking at different forms of damage. The tunnel assay used the terminal TDT transferase to label 3 delta N free ends of DNA, resulting in high labeling sperm with fragmented DNA. The COMET assay has a unique feature that can distinguish between single and double-stranded DNA breaks when it's performed under alkaline or neutral conditions. It is based on nuclear decompaction followed by electrophoresis and visualization of individual sperm. In other words, it's complicated. Sperm chromatin structure assay, SCSA, which is one that I've been involved with since the early 80s, looks at sperm chromatin dispersion. There's an error there. And is based on the detection of sperm for fragmentation based on the denaturing capacity of the sperm chromatin. And I think I'll just skip over that. This is basically another great article in RBM Online. So it's open access, so everyone should read it. And it's a really quite an important article for scientists to read, looking at all the different enzymes that are in the oocyte that can repair all the nicks and breaks, etc., that are found in sperm DNA fragmentation. But basically, the repair potential is highest in the zygote and diminishes very quickly as you go on. In fact, I understand most of the repair work is actually done at this stage. So they find indicating that oocyte quality, which diminishes with age, significantly impacts the efficiency of DNA repair process, thereby influencing embryo viability and pregnancy success. While younger oocytes can partially compensate for sperm DNA fragmentation, aging oocytes show reduced repair capacity, contributing to poor reproductive outcomes. These insights emphasize the need for novel reproductive strategies aimed at enhancing oocyte quality and repair efficiency. But the thrust of this paper is pointing out that even though the oocyte has considerable capability to repair small nicks and breaks in the sperm DNA, particularly sperm DNA, that repair falls away very quickly as the woman becomes older. And therefore, and unfortunately, older women usually have older male partners, but they have more sperm DNA damage. And so the problem with older women both is it's got a lot to do with the inability of those eggs to fix broken sperm. So if there are different types of sperm DNA damage tests and the ability of the oocyte to repair them may also differ, are there any tests, are any of the tests a marker of more general sperm damage over lots of sperm? Are we taking the DNA fragmentation test too literally? So not only do we have a situation where we have different tests that apply in different ways with age of the guys, there's also the effect of the oocyte to repair them, which will have some impact on trying to work out whether the test has got any potential to influence the euploidy rate and pregnancy rates. It's all too really very complicated. But as the point I wanted to make here is, are any of the tests a marker of a more general sperm damage, not specifically just how the sperm wrapped up or breaks? Are we taking DNA fragmentation tests too literally? After all, one only needs a few sperm from 100 million or so for insemination. How hard can it be? Okay, so after a long preamble, let's get back to the paper that initially prompted this video. The answer was to quantitatively evaluate the association between sperm DNA fragmentation and embryo euploidy rates in IVF, doing a systematic review and meta-analysis. And they're kindly to publish a flowchart of their meta-analysis. And the thing that struck me was they started doing a PubMed search and Embase and Scopus and Cochrane Library, etc. They found 1,356 articles and only found 6 they could review. So there were 1,350 articles published using sperm DNA fragmentation and IVF that were totally useless for this meta-analysis. But they did limit it to only sperm using the SCSA test, which is looking for chromatin, how well the chromatin has been wrapped up. And it goes to show how much useless information is published, and little wonder people find it confusing. It's easy to discredit sperm DNA fragmentation, ignore it, and just say it doesn't make any difference. Let's not worry about it. Don't bother testing it. And I think they're wrong. So what the authors of this paper did, they looked at 3 levels of DNA damage in the 6 papers: low level, more than 15%. That's basically almost above background level. Between 15 and 30%, which is a lot of people talk about 20% DNA fragmentation, which fits in this moderate level, and high levels of fragmentation, more than 30, which in fact I have always used myself at a clinical level. So a series of forest plots comparing less than 15 to more than 15, and of these had 4 papers that could apply for this. Excuse me. And basically there was no difference in the odd ratio of the effects of fragmentation on the euploidy rate. Excuse me. Better have a cup of coffee. Okay, if you compare 15% to this middle level, once again, there's a slight shift to the right, but there's really no significant bias in the odds ratio. Therefore, you can't distinguish between less than 15 and a moderate level of DNA damage. If you compare very low levels of DNA damage to a high level, the odds are starting to become more of a significant relationship, but still not at a significant level. You see here there's 3 papers, one showing a definite relationship, one showing one is slightly biased towards it, but there is actually no significant difference in the odds ratio. Likewise, if you look at low levels of damage to like the moderate level to high level, once again, the odds ratio is moving towards an effect, but not to a significant effect. And finally, if you get to more than 30% to less than 30%, you start to find a significant difference. So at this level, the authors concluded that there was a significant relationship between sperm DNA fragmentation and the euploidy rate if you use 30% as a cutoff for a SCSA test. And once again, this is not talking about the age of women, as far as I understand. I don't think the age of the women were part of these 5 studies. So they concluded, this systematic review and meta-analysis provides the first comprehensive quantitative synthesis of the relationship between sperm DNA fragmentation and embryo euploidy rates in IVF cycles. Amongst the 6 eligible studies of 1,500 odd cycles, they reported they found an elevated sperm DNA fragmentation was significantly associated with a reduced likelihood of obtaining euploid embryos when a higher threshold of more than 30% was applied compared to everything else. I just want to quickly run through the different papers that were part of this meta-analysis. So 16, 27 and 28, these results align with recent evidence showing that high sperm DNA fragmentation may compromise embryo chromosomal stability through mechanisms involving oxidative damage, defective chromatin package, and impaired paternal genome repair capacity. In these 4 papers, the authors concluded the inability of mature sperm to repair DNA lesions means that the oocyte must compensate. However, oocyte repair capacity is finite and declines with maternal age. And I think we're starting to get to the very heart of the matter here. And then looking at one paper by Fu, etc., This is consistent with the findings of Fu, et cetera. Well, their findings were consistent with that of Fu, et cetera, who reported that elevated sperm DNA fragmentation was significantly correlated with an increased chromosomal euploidy rate in miscarried conceptuses from women of advanced maternal age undergoing fresh transfers with an optimal SDF threshold as low as 8%. Such results highlight the potential synergistic effect between paternal DNA damage and diminished oocyte repair capacity, and may partly explain the age-dependent susceptibility observed in some studies. So, getting back to the final paper that was quoted from Braga, etc., a high sperm DNA fragmentation greater than 30% increases the likelihood of miscarriage in ICSI cycles, an effect that may potentially be magnified by the presence of oocyte dysmorphisms. Okay, all right. So getting back to the original paper, this study findings suggest that elevated sperm DFI as measured by the SCARSA test. Oh, sorry, this was one paper that was the outlier. I apologize. This is an outlier one looking at the SCARSA test, and in this group, published in 2025 in Andrology, this study found that the elevated sperm DFI as measured by SCARSA does not significantly influence euploidy rate of viable blastocysts with couples maternal age, whereas maternal age remained the predominant factor influencing embryo development. So I think in here they're saying, in this paper, they looked at maternal age, which is clearly related to the euploidy rate, and they found that the elevated sperm DNA fragmentation did not add significantly to that observation, or did not independently predict it. So in this study by Ping, association of embryo euploidy and sperm DNA fragment in unexplained recurrent implantations for patients, the component of now this is a bit complicated, however, the component of aneuploidy embryos was significantly higher in high DFI groups rather than the medium group and the low group. The miscarriage rate for high DFI groups and the medium group was significantly higher than the low group. There was no difference to be found between regarding fertility, good quality embryos, pregnancy rates, live birth, and newborn rates amongst the three groups. So it's all rather complicated. But once again, I think we're looking at what's been shown for a long time is that samples with high DFIs result significantly in increased miscarriage rates. And miscarriage rates are a reflection of aneuploidy rates. So they concluded the sperm DNA damage is associated with blastocyst aneuploidy and miscarriage rates in unexplained recurrent infertility patients, embryo selection by PGTA, and efforts to decrease sperm DFI before ICSI should be considered for those male patients with a high DFI. And how do you know if someone's got a high DFI unless you test for it? So all the clinics that are doing IVF and ICSI are not testing for sperm DNA fragmentation, particularly if the women are over 40, I think not doing any, doing a disservice to their clients. And one last paper from GATT, the total number of biopsied embryos that are in groups 1, 2, and 3, PGS for 24 chromosomes revealed comparable euploidy rates of 45, 46 to 50% with similar morphology and classifications. And in this group, that they couldn't find any difference was found regarding pregnancy rates or pregnancy loss rates. It seems that DFI doesn't correlate with blastocyst aneuploidy morphology rate. And once again, this is just another paper which is an outlier. Just need to go back. So in this one, the effect of sperm DNA fragmentation on whole the segmental chromosome aneuploidies in human embryos. So compared to that in the low DFI group, segmental chromosome aneuploidy rates were significantly higher in the high DFI groups. The whole chromosomal embryonic aneuploidy rate of paternal origin was significantly higher in cycles with high DFIs than low DFIs. However, the segmental chromosome aneuploidy of paternal origin was not significant between the two groups. In conclusion, our results suggest that high sperm DNA fragmentation was associated with the incidence of segmental chromosome aneuploidy and increased paternal whole chromosome aneuploidies in embryos. So segmental aneuploidy. Contemporary interpretations of aneuploidy in embryos also include segmental losses and gains where there are deviations in the normal copy number of subchromosomes, such as affecting severe megabases. So this is actually from this paper. Segmental aneuploidies are mainly believed to originate from mitotic errors during early cell divisions of the forming embryo. The development potential is associated with weakened cell cycle checkpoints and lax DNA damage repair and double-strand breaks due to rapid proliferation. Approximately 16% of IVF-generated blastocysts contain segmental losses or gains. Interestingly, of these segmental aneuploidies, only 5% arise at the germ cell level, derived errors largely from paternal contribution. Okay. So getting back to the original paper, I think they have concluded that overall elevated sperm DNA fragmentation, particularly above 30%, is associated with reduced likelihood of obtaining euploid embryos in ART cycles, suggesting a potential threshold-dependent effect of sperm DNA integrity on embryo chromosomal normality. These findings support the integration of SDS assessment in the elevation of selected couples, especially in cases of recurrent ART failure or advanced maternal age. And I just wanted to have one last slide. I made another video on this, is looking at sperm DNA fragmentation and phospholipase C efficacy in fertile couples. So this is not an open access in current issues of molecular biology by a group of, I think, Algerian authors. And the important thing the reason we're including this right at the very end of the video is that if you look at the percentage of sperm in fertile and infertile patients. Basically, fertile, because they have a high level, most sperm, 65%, have detectable levels of PLC in the sperm head, whereas in fertile patients, it's only around about 20%. And it's roughly the inverse of where there's sperm chromatin decondensation and fragmentation. And the reason that's important is because phospholipase C is the trigger that sperm releases to cause these release of calcium in from the endoplasmic reticulum in the cytoplasm, and these pulses that vary in intensity and frequency. And the intensity and frequency will determine the rate of cell division. And the rate of cell division will then, in fact, the rate of mitotically induced aneuploidies. The comparison of mean percentages of spermatozoa presenting with phospholipase C zeta, DNA fragmentation, chromatin decondensation between fertile and infertile couples is that they're different. So just to finish, I like these pictures. This is a really good picture of a sperm swimming up the fallopian tube. This is a picture of one butterfly in amongst many. I couldn't find one sperm in amongst many, but I'm looking. But basically, when you're doing ICSI, you're trying to pick from all these motile sperm one to inject. And if all these sperm have varying degrees of DNA fragmentation, but more importantly, DNA damage, different gene constructs, etc., like phospholipase C, how do you know which ones to select? So my comments on this whole paper is this. If you see sperm DNA fragmentation as a metaphor for sperm health, of all the sperm in the sample, then even normal sperm may harbour some degree of segmental aneuploidies, nicks, breaks, poor chromatin, etc. The damage may have less to do with DNA and may be more widespread, and phospholipase C is a fantastic example. This study suggests that it may have the capacity to influence or directly or indirectly the euploidy rate. We need to recognise that this will give rise to various forms of aneuploidy, and to try and improve sperm selection using microfluidic processes, especially when the age of the oocytes will impact on their ability, is important. And if you don't do this, the only way you can really affect this issue is to use younger donor eggs or younger donor sperm, which is what we're trying to avoid. So that, my friends, is a lot for the week. My name's James Stanger. I'm an embryologist with 50 years experience. I'm not a medical person, and this information is not medical advice, but it may help you in knowing what questions to ask about your treatment with your doctor, and maybe to help in understanding the advice that you receive. If you like what we do, click subscribe or leave a comment and click like. And just lastly, just one little plug for my other business. My Fertile website is an online EQA program, a training program in IVF, in andrology and embryology. And if your staff are enrolled in this, from the client's point of view, they may benefit from knowing that the skills of the laboratory staff are on constant review and assessment. And the EQA schemes include andrology and embryology. They can also train as professional, small clinical, multi-lab clinics used for training, competency, or accreditation. That, my lot, they say, is your lot for the week. I'd like to thank you for your attention. I know IVF is a personal journey that involves the action of many different techniques and many different people, and it helps to understand all aspects of this journey. I hope this information may be of value. If you like the information I provide, please tell others, and we'll see you again. Thank you.